This is a story about how scientists who poked around Yellowstone's hot springs changed the fields of biology and medicine forever. In 1985, an amazing new experiment called PCR had just been invented. The polymerase chain reaction allows scientists to take a sample of DNA, which contains all of the genes of the organism from whence it came, and isolate a specific gene from that and make millions of copies of it. The enzyme that makes all of these DNA copies is called DNA polymerase, and the OG polymerase used in PCR came from E. coli, the bacteria that live in our guts. But PCR involves like 20 cycles of heating all the way up to almost boiling and then cooling again. And E. coli isn't super happy above our body temperature, and neither are its enzymes, so DNA polymerase would just kind of melt. That meant you needed to add new polymerase every single PCR cycle. So to do one of these experiments, you had to babysit it for hours, and there were like 20 plus chances to mess it up somehow. But let's rewind. In 1969, Brock and Fries published a paper describing a new species of bacterium, Thermus aquaticus, that thrived in the hot springs of Yellowstone National Park. It was the first of its kind to be isolated and grown in the lab, and the paper is mostly like, hey, it's so neat, we figured out how to do this really cool thing, there's some cool stuff about this bacterium. 7 years later, Alice Chien successfully isolated DNA polymerase from Thermus aquaticus and was like, hey, I don't know, but this seems like it could be really cool for some like gene synthesis experiments or something. PCR hadn't been invented yet, but they were kind of in that general territory starting to figure stuff out. And then within a few years of the invention of PCR, scientists were using this heat-tolerant DNA polymerase, which they affectionately called TAC, if you stick the names together. Thermus aquaticus, TAC, yeah. And TAC immediately revolutionized PCR. You could just add all of your ingredients and forget about it because the polymerase would last all 20 plus cycles. Today, PCR is the backbone of literally all molecular biology and medicine. The entire field of genetics wouldn't be possible today without PCR. And in medicine, oh my god, paternity tests, gene therapies, COVID tests, vaccine development, PCR is key to each and every one of them, and it's only possible at this scale because of TAC polymerase. But the original 1969 paper about the weird little bacterium, that research was funded by a 3-year grant from the National Science Foundation, the federal agency that funds discovery science, science that doesn't have an immediate application at the time, but is figuring out fundamental mysteries of our universe, like what the fuck is living in hot springs? And then sometimes that discovery science goes on to change the world. The NSF is going to be cut by 60% if the 2026 budget passes, which will destroy the future of discovery science here. So yes, this is a sneaky edition of what the funding. Government funding for discovery science is essential. Industries and private funders are not going to support stuff like this if it's not guaranteed to make them money. These scientists were just trying to figure out, hey, what's in the hot spring? They didn't have an agenda. PCR is what it is today because the NSF invested in scientists' need to study the weird and wonderful things in our beautiful world. If you think the only reason the NSF should exist is because it funds things that could have applications in the future, fine. But personally, I think it's magical that humans have the ability to study and understand and describe all the weird little guys on this planet and beyond. And that's enough for me. This week, scientists are advocating for public research funding using the hashtag #RootingForScience. So if you want to see more stories like these, check it out.